Hi folks, and welcome back to our second renal physiology topic. And believe it or not, we're going to talk for the next 40 minutes on glomerular filtration. So it seems like a little minor point, but for the kidneys it's actually a major thing, because that's really what they do. They filter the blood. So we're going to have a look at how does the kidney filter the blood and what are the things that control it, because you'll learn in today's session that there's a number of mechanisms that are designed in our body to control that glomerular filtration rate. So we're going to cover some glomerular filtration, really, and that's today's plan. We'll quickly talk about the nephrons and what they do in terms of being the functional unit of the kidney, and then we'll spend the rest of the time on what this glomerular filtration is. And then the last bit is how we regulate glomerular filtration. And you'll see we're using abbreviation there of GFR. That GFR is glomerular filtration rate. So it's really how we regulate glomerular filtration. Okay. All right. Now, remember the last session we talked about the nephrons. And the nephrons we said were the functional unit of the kidney, because each nephron that we have in our kidney performs the three basic things that the kidney as a whole does. A nephron filters, a nephron reabsorbs, and a nephron secretes. And what's left after that process is what is urine. It's what is excreted as urine. So the nephron itself does the things that the kidney does. Glomerular filtration, so it filters the blood. What it wants back from the filtrate, it has to reabsorb, so that's tubular reabsorption, and then what it also wants to get rid of but didn't get a chance to filter, it can secrete as well. So those three processes are demonstrated very simply in that diagram there. So there we have a very, very stylized diagram of a nephron, don't we? Over on the left here, here's our glomerular capillary network, and here's our glomerular capsule or Bowman's capsule that receives the filtrate, and this is where we have filtration. So number one represents filtration. All that filtrate that enters into the Bowman's capsule or glomerular capsule travels down our renal tubules and collecting duct. What we want back from the filtrate and put back into the blood, like water and solutes and glucose, amino acids, all the good stuff that we want back, we have to reabsorb. And that's this number two process here. That's tubular reabsorption. And to a much, much lesser extent, so this isn't a major thing that the nephrons do, but it does actually happen. If some of these things that we want to get rid of from the body didn't actually get filtered back here at the glomerular capillary, we can actually move some of those substances from the blood into the tubular network of the nephrons. So that's number three there, tubular secretion. This is particularly important for things like hydrogen ions. When our kidneys try to regulate our acid-base status, the kidneys can secrete hydrogen ions from the blood and put them into the renal tubules, so we excrete the hydrogen ions in the blood. But they're the three processes that the nephron does, and it's really the three things that the kidney does in terms of filtering the blood. And of course, the balance of what's left after filtration, reabsorption, secretion, whatever's left after that is urine. So it's the job of one nephron that does the job of the whole kidney. So it's the millions of these nephrons that collectively does the job of the one kidney. Okay? So that purple thing just describes that sort of concept there, that the rate of excretion of any substance, so the rate at which it's appearing in the urine over here, is really a function of the rate at which it's filtered, the rate at which it's secreted, minus its rate of reabsorption. And that gives us an indication of how fast we're excreting something. So that's the nephron. Now the rest of the session is devoted to glomerular filtration. you might think, how can I possibly talk for the next 30 minutes on this one thing? Well, hold on and we'll go through glomerular filtration. What is it, how it's regulated, and what controls that regulation. So, here's some numbers. You don't really need to know these numbers, but it gives you an idea of the scope of things. Glomerular filtration is the formation of protein-free filtrate of plasma across the glomerular membrane. So that tells us something. It tells us that there's a selectivity there. So the things that get forced out of the glomerular capillary are only small things. So water, the things that are dissolved in that water, like solutes and amino acids and glucose, very small organic molecules can get through that filtration membrane or through that filtration barrier. It's protein free. If you have protein appearing in your urine, you have problems with your kidneys, and it's actually one of the things that are used to diagnose kidney disease, putting a dipstick in the urine and seeing if there's protein there. So let's have a look at some other general things. Glomerular filtrate. How much glomerular filtrate do we produce at the million nephrons in each kidney? So 2 million nephrons, how much filtration is there? It's quite a lot. It's about 180 litres a day. So when we talk about glomerular filtration, it's a lot of fluid that's created. 180 litres, okay? And we have to return that back into our bloodstream. We obviously do that because we would be filtering our entire blood volume within 2 minutes, and we would, if we didn't get it back, we'd be completely dehydrated. So obviously this process of glomerular filtration is a big process, but an even bigger process, which we'll talk about in the next session, is that reabsorption process. We've got to get it all back. Okay? So if you do all the numbers of 180 litres a day that's been filtered, what that actually adds up to, let's try and do the numbers, it's about 12 times per day that we filter our blood volume, okay? So the kidneys are really, really efficient at filtering our blood, aren't they? They don't just filter it, once or twice a day. We filter the entire blood volume each day, 12 times. Okay, so that gives us an idea of the scope of things. So, what is this filtration? How does it happen? Okay, we touched upon it in the previous Camtasia, but there is what we can call a filtration membrane, which is made up of the glomerular capillaries, which have fenestrations in them, or little holes, which are surrounded by the little podocytes, which have the special feet that interlock with each other and produce these filtration slits. Acts as another filtration barrier. And we also have what's termed a basal lamina, which is a sort of barrier that sits on the base of our endothelial cells and actually acts as another filtration barrier. So we've got three things. We've got the glomerular capillaries, the basal lamina, and the podocyte feet, which form filtration slits, and that's our filtration membrane. So it permits water and small solutes through, and it prevents the filtration of plasma protein, red blood cells, white blood cells, and platelets. So when you have blood appearing in your urine, when you have white blood cells appearing in your urine, or platelets appearing in your urine, or plasma appearing in your urine, it indicates that there's something wrong with the filtration membrane in your kidneys. It could also mean you've got a urinary tract infection, particularly if it's just white blood cells that you're seeing. But in terms of protein appearing in your urine, it can really indicate there's something wrong with your filtration membrane itself. It's been damaged and letting things through that it shouldn't let through. Okay, we talked about what consists of that filtration membrane. It's the glomerular endothelial cells and their holes in it, the fenestrations. It's the basal lamina that sits sort of at the base of the endothelial cells, and it's the filtration slits that are formed by our podocytes. Okay? And you might think, well, we've only got 1 million nephrons. How do 1 million tiny little nephrons filter 180 litres a day. It's because the volume is so big, because the surface area is so large. Even though we've got 1 million tiny little nephrons, the surface area of that filtration membrane is massive. So we have a very, very large surface area, and we actually have a high pressure that's driving that filtration. And we'll get to talk about that pressure soon. So there's our three things that make up the filtration membrane in a diagram there. Number one is our holes, or our fenestrations. Number two, this layer here, as we said, is the basal lamina, which sits at the base of the endothelial cells. And the third one are the podocyte feet, which form slits. They're our three components of the filtration membrane. Okay, so how do things get through? How do things get filtered? They get filtered by pressure. And you might think, oh, that pressure is the blood pressure. Well, essentially you're correct. So the pressures that determine how much stuff gets pushed through our filtration membrane is essentially our blood hydrostatic pressure. They call it hydrostatic pressure because blood is a liquid, meaning it's hydrostatic pressure. But there are two other pressures involved, which we'll deal with shortly. But essentially, if you want to hang on to the concept of we filter at the glomerular capillary through the filtration membrane because of we have a blood pressure, that's kind of correct. But let's qualify that and talk about two other pressures in there as well. There is a capsular hydrostatic pressure, and that actually opposes filtration. This is the pressure of the fluid that's already in the Bowman's capsule, or already in the renal capsule. So there's fluid that's been filtered, and this actually opposes the pressure of the fluid coming into the capsule as well. And the other pressure that we should think of, and we've come across this pressure before when we talk about capillary filtration at the tissue capillaries, it's the colloid osmotic pressure. And this is due to the plasma proteins that don't get filtered. Remember, we talked about the filtration membrane as being a selective thing that only lets small things through. It doesn't let big proteins like your big albumins and your big immunoglobulins, and doesn't let red blood cells or white blood cells or platelets through. These are big proteins and big things that exert what they call a colloid osmotic pressure. And they have the effect of wanting to draw fluid back into the glomerular capillary. So they oppose filtration. Either way, we know we have a net filtration pressure, which is positive. So that's this last thing here, net filtration pressure, or NFP, is the hydrostatic pressure. That's the largest pressure that promotes filtration. But we've got to subtract the capsule hydrostatic pressure and also the colloid osmotic pressure. And whatever the difference is, that's what we're left with. And it turns out that it's about the equivalent to 10 millimeters of mercury. So our blood pressure is, say, 150 on, or 120 on 70, okay? 120 on 70. So it's about a tenth of your diastolic pressure. So 10 millimeters of mercury. And that's the pressure that forces all that ultrafiltrate through into all our nephrons. So we're really just looking at a diagram there of those filtration pressures. So number one is the blood pressure, glomerular blood hydrostatic pressure. It's 55 millimeters of mercury. It's 55 because we're looking at arterial blood pressure. So we've started right out at the heart at, say, a systolic pressure of, say, 120. But when we reach the kidneys, it's dropped right down to about 55 at the level of the arteriole as we move away from the heart. So that's 55. That's pretty big, isn't it? But we actually have a hydrostatic pressure in there, and again, that's a water pressure. This is the ultrafiltrate that's within this capsule here. And because there's water in here, it's going to exert an effect, and it actually opposes the hydrostatic pressure, and it's got a value of about 15. So it's minus 15, if you like. The other pressure in here that's quite significant that opposes the hydrostatic pressure of the blood is the colloid osmotic pressure. This is due to those big plasma proteins, albumins, immunoglobulins, and protein components of the cells. They want to actually draw water back into the glomerular capillaries, so they oppose that hydrostatic pressure. And we can think of it as being about -30. So our net filtration pressure is really just a function of 55, taking away 15 for the hydrostatic in the capsule and 30 for the colloid osmotic, and we're left with a difference of 10. So take-home message, net result of the pressures that exist at the glomerular capillary is we have a net filtration pressure of 10. We have a huge surface area for filtration, so we have a huge volume of filtrate that's formed by glomerular filtration, up to 180 litres a day. Okay, so what is that rate? So we've got 180 litres a day. What does that equate to over a minute? Because we now want to quantify that to a number that is clinically meaningful and goes a long way to telling us what a kidney is doing in terms of function. So they quantify this glomerular filtration concept into a glomerular filtration rate, or a GFR. And it's the amount of filtrate that's formed in all of our little nephrons in both kidneys in a minute. And if we work that out, it turns out that in a normal adult male of sort of middle age and normal weight, it's about 125 mils per minute. Now this tells us kidney function. So when you come to look at people's blood work and they're assessing kidney function, you'll see that they calculate out GFR. They calculate out GFR by measuring the concentration of a particular thing called creatinine. But they basically quantify glomerular filtration rate by working it out to mils per minute. And that tells us kidney function. If that number is very, very low, it indicates a very bad kidney function. So it's used as a way of quantifying kidney function, and it's used actually clinically to diagnose chronic kidney diseases and the stages at which they present. So the stage 4 and stage 5 have glomerular filtration rates, or a GFR, of between 10 through to 25 mils per minute. It's telling us that their kidney is only filtering 10 to 25 mils per minute, whereas normally it should be about 125. Okay? So, glomerular filtration rate, it's about 120 in us. Gets worse as you get older, but it's all calibrated with respect to age and gender as well. But why do we worry about glomerular filtration rate? Why do we worry about quantifying it? Well, if it gets too high, if our glomerular filtration rate gets too high, we filter so much fluid and so many solutes and nutrients that we can't reabsorb it all fast enough, and we'll actually lose a lot of the good stuff in our urine. If it's too low, and that's essentially what happens in chronic kidney disease, or CKD, what happens in chronic kidney disease is the glomerular filtration rate declines too much, and you get the build-up of waste products in the body, and particularly those nitrogenous products like urea and those sorts of things build up, cause all sorts of problems with body function. Okay, so let's start to think about glomerular filtration rate, and it's kept in a fairly narrow range. Too high, we lose all the good stuff. Too low, we accumulate all the bad stuff. We want to keep our glomerular filtration rate at about 125 mils per minute. Now there's some of the numbers that we were talking about before, some of those big numbers that we're talking about. Amount of filtrate, we filter about 180 litres of water or plasma. We filter 180 litres of plasma a day. and we reabsorb about 178 to 179 litres of that. So we lose about 1 to 2 litres of our water from the plasma in one day. Okay, it seems like a lot that we lose, but look at how much we filtered. We've filtered 180 and got back about 178 to 179. Okay, very, very minimal amounts of protein appearing in the urine. Virtually no glucose. That's the filtered amount of glucose that you would normally filter in a day, is about 162 grams of glucose. I think 1 teaspoon is 5 grams. So what's that, 5 into 160? You do the numbers. It's quite a number of teaspoons that we filter at our kidneys, and we get it all back. We don't have glucose appearing in our urine. Urea is a waste product. We filter about 54 of that, and we get rid of about 30. So we actually get some urea back, don't we? We filter for 54, and we return about 24 grams of that. Okay, all right. So next thing we're going to talk about is how we regulate GFR. We've got to regulate it because, as we said, too high we lose stuff in our urine, too low we accumulate waste products. So we regulate GFR, and most of the time we don't even know we're regulating our GFR because the kidney does it itself. Okay, so we're going to talk about how the kidney itself regulates GFR, and then when we're in trouble, our nervous system and our hormones can start to regulate GFR. But this is only really when we're not worrying about GFR, we're worried about maintaining blood volume and therefore blood pressure. So there's kind of two things that we need to worry about with GFR. There's what the kidney does to regulate its own GFR, and that's to maintain the right amount of filtering. But there's the nervous system and the hormones that can override all that when we're worried about maintaining blood volume and blood pressure. Then all bets are off and we don't worry about GFR, we worry about blood pressure. So let's go through those concepts there. We've got 3 principal mechanisms which we'll deal with. Renal auto-regulation. It's how the kidneys regulate their own GFR, keep GFR constant, keep it at the right level. There's the neural regulation. This kicks in when our autonomic nervous system starts to regulate blood flow and GFR. This tends to happen, as we said before, when we're not worrying about keeping GFR constant, but when we're worrying about trying to keep our blood pressure constant. And then there's hormones that belong to the renin-angiotensin-aldosterone system, and hormones that are related to the other side of the coin, the atrial natriuretic peptide. So let's go through those 3 principal mechanisms, and there's a couple in the renal autoregulation that we'll talk about. So how does the kidney regulate its own GFR? One of the ways in which it does it is through a myogenic mechanism. Myogenic means, myo means muscle. Where is the muscle in the kidney? The muscle in the kidney is in your arterioles. And it's just a little local reflex that every little afferent arteriole has. And if there is an increase in systemic blood pressure, that means there's an increase in systemic blood pressure. That means there will be an increase in the filtration pressure. That's filtering filtrate across our glomerular capillaries and into our renal capsule. If we don't do anything about that, the glomerular filtration rate is going to go right up because the filtration pressure of the blood, or hydrostatic pressure, has gone up. So what happens? Because there's an increase in pressure, there's the afferent arteriole stretches, and in response to that stretch, our afferent arterioles actually contract to reduce the diameter of our arteriole, and that actually returns the blood pressure within our little glomerular capillary back to normal. So we don't have a big change in GFR despite us having a big change in systemic blood pressure. This is really what controls our GFR when we go off and start exercising. as you would have done in that first lab, when we exercise, our blood pressure goes right up because we've got an increase in stroke volume, increase in heart rate, got a massive increase in cardiac output. That's going to drive a big rise in blood pressure. But we actually don't start filtering more blood across our glomerular capillaries because we've got such a huge rise in blood pressure, because our little afferent arterioles have all constricted down to stop that rise in filtration that's happening at each nephrons. So that's our first mechanism. It's a myogenic mechanism. It's intrinsic to our arterioles. They constrict in response to stretch, and it's the stretch from increasing blood pressure that makes them constrict up and return GFR back to normal. Now, here's another one, and we've talked about this system before. This renal autoregulation of GFR and the tubuloglomerular feedback. It involves the juxtaglomerular apparatus. So there's two big words, two big terms, and they're all related. Tubuloglomerular feedback involves the juxtaglomerular apparatus. Okay? Now, we talked about the juxtaglomerular apparatus previously, didn't we? It's that system whereby the afferent arteriole comes very, very close to the ascending loop of Henle. Okay? And if the ascending loop of Henle senses that there's a big increase in flow past it, what it does, it releases factors from the macular denser cells that vasoconstrict the afferent arteriole. And if we vasoconstrict our afferent arteriole, it reduces our GFR back. Okay? So essentially what we've got is the same pattern. If we've got an elevated systemic blood pressure, it has the potential to raise GFR so that there's too much fluid going past the loop of Henle. And that gets sensed, and the macular denser cells release factors which make the afferent arteriole vasoconstrict, returning our GFR back to normal. Okay? So that's our renal autoregulation. And we've got that sort of flow diagram there. And this relates to the tubuloglomerular feedback that we've just talked about. So let's follow this from the top. We've got an increasing glomerular filtration rate. This could be from an increase in systemic blood pressure. That is sensed by macular denser cells, which detect the increase in sodium and chloride and flow up past the ascending loop of Henle. That then tells the juxtaglomerular cells, the macular denser cells, to release factors which essentially cause the afferent arteriole to constrict. And if we're constricting the blood flow, we are then decreasing the GFR. Okay? So there's vasoconstrictors and vasodilators involved in that process. They've actually talked about the decreased secretion of nitric oxide, but there's vasoconstrictors and the nitric oxide involved. But the net result is that little system of sensing flow results in decreasing our GFR. Okay. Now, on to neural regulation of GFR. And as we said before, the only nerves that go to the kidneys are the autonomic nerves, and it's the sympathetic division of the autonomic nervous system. There is no parasympathetic supply to the kidneys, it's only sympathetic. So it's the blood vessels of the kidneys are supplied by the sympathetic fibres, and they release noradrenaline to cause vasoconstriction of our afferent arterioles. Okay? So vasoconstriction of our afferent arterioles, what's that going to do to GFR? It's going to decrease it, isn't it? So let's have a look. There's our little sympathetic nerves coming in, innervating our afferent arteriole, and if we vasoconstrict our afferent arteriole, we're going to be reducing the flow through here, reducing the pressure through here, getting less filtrate through here. So that's essentially what happens. At rest, we've got maximal dilation of our afferent arterioles. So it's the renal auto-regulation that prevails, the myogenic mechanism and the juxtaglomerular apparatus and the tubuloglomerular feedback. That's what at rest is going on. So with moderate sympathetic stimulation, so with a little bit of stress, for example, not too much stress, but a little bit of stress, moderate sympathetic stimulation, both the afferent arterioles and the efferent arterioles constrict equally, decreasing GFR equally. So we're actually retaining more fluid in our blood rather than filtering it through our kidneys. Okay? But what happens during exercise, for example, or extreme amounts of blood loss? We get a lot of sympathetic tone going to our afferent arterioles, and that causes a huge amount of vasoconstriction of our afferent arterioles, and it reduces GFR significantly. And what does that do? It reduces filtration, and it will therefore reduce the amount of urine that we're producing. And that's really an awesome thing. We really don't want to be having the urge to urinate when we're exercising. We want to divert all of that fluid away from our kidneys and have it in circulation so it can be oxygenated and go to all our exercising muscles. Okay? It's also awesome if you're bleeding to death, if you've got a hemorrhage. You don't want your kidneys to have 120 or 180 litres of fluid a day going through your kidneys. You want to try and keep it in your cardiovascular system to maintain your blood pressure. So they're two really good things that the sympathetic nervous system does in order to maintain blood volume if you're hemorrhaging, and also to divert blood away from the kidneys to the tissues if you're exercising. Okay. The other thing that we can do to regulate our GFR is biohormones. And one hormone increases GFR and the other one decreases GFR. So the one hormone we should worry about that increases GFR is called atrial natriuretic peptide, or ANP. And it's released by the heart. So the heart actually produces a hormone, and it's produced by the atria. And the atria produce ANP in response to being stretched. And that stretch happens when our blood volume increases and increases and increases, and it stretches the atria. And the atria release a hormone, ANP, and that increases GFR, because increasing GFR will then allow more water to be filtered through into the nephrons, and we'll get rid of excess water that way. Okay. The other hormone that's very, very important, and it has a role in reducing GFR, is called angiotensin II. In itself, it's a very, very potent vasoconstrictor. It narrows afferent and efferent arterioles and has a very, very potent effect on reducing their diameter, just like the sympathetic nervous system, reducing their diameter, and it narrows the afferent and efferent arterioles and reduces GFR. Okay, so we're conserving water when we've got angiotensin II around, because we're narrowing our arterioles down and we're not doing as much filtering. So angiotensin II and sympathetic nerves stimulation during sympathetic nerve stimulation or through hemorrhage actually go hand in hand. Angiotensin II and sympathetic nervous system are all trying to do the same thing: reduce GFR to conserve blood volume, or to divert blood away from the kidneys and to muscles itself. Okay, so here's a summary which we're going to break down, and it actually summarises all the things that we've talked about. So we're going to break this great big table, which doesn't make a lot of sense on its own because there's lots of words and arrows going everywhere, but it's just a summary of actually what we've talked about in this Camtasia. Okay, they're all responses to changes in blood pressure, because it's the change in blood pressure that has the potential to change GFR. And how do we regulate that change? So let's do the first one. It's the myogenic regulation of GFR. Okay, So this is a function of the arterioles sensing stretch or lack of stretch. So in this one, decreased systemic blood pressure. The decrease in blood pressure in the afferent arterioles decreases our GFR. Okay? So, if there's a decrease in blood pressure, that means there's now less stretch on the smooth muscle walls in the afferent arterioles. So in response to less stress, what do? They vasodilate, and that increases the flow through those arterioles, leading to an increase in GFR. So we've reversed the decrease in GFR by increasing the GFR back to normal. And it's all through that changing in the stretch. We first talked about the example with an increase in stretch. This is an example where we're talking about a decrease in stretch. And the result is we've returned GFR back to normal through a myogenic mechanism of sensing stretch or lack of stretch and changing the diameter of the blood vessel. The other one we talked about was tubuloglomerular feedback, again intrinsic to the kidney, autoregulation. With a decrease in blood pressure, we have a decrease in GFR. If we have a decrease in GFR, the filtrate or the rate of flow of fluid through the ascending limb of the loop of Henle is going to go down. The macular denser cells can sense that change in flow rate, and when there's a decrease in flow, we have vasodilation of the afferent arterioles. We have vasodilation of our afferent arterioles. That's going to return our GFR back to normal in response to that initial decrease in GFR. This is the hormonal regulation of GFR by angiotensin II. We're just going to follow the right-hand side of this diagram. We're not going to worry about the left-hand side just yet. So in response to a decrease in blood pressure, we have the granular cells release renin, and that activates the renin-angiotensin-aldosterone system. That causes the production of angiotensin II. If we follow the right-hand side, angiotensin II causes vasoconstriction of the systemic arterioles to increase peripheral resistance, and that will cause an increase in systemic blood pressure, and that will actually cause a return of our GFR, okay, in response to a drop in blood pressure. Okay. All right. Next one on our list is the neural regulation of GFR, okay, and this is again in response to a decrease in blood pressure. We have inhibition of the baroreceptors in the blood circulation. So remember way back we talked about the baroreceptor reflex. Baroreceptor reflex is a response that is within us to sense stretch of our aorta and stretch of our carotid arteries. And when we've got a drop in blood pressure, the baroreceptors detect that. And the baroreceptors then fire off and tell the sympathetic nervous system, I've got a drop in blood pressure. What that does is the systemic nervous system responds and causes vasoconstriction of the systemic arterioles to increase peripheral resistance. Okay, we're increasing our peripheral resistance, which means we're vasoconstricting. Now remember, this is the difference between the hormonal and the neural regulation of GFR. This is changing GFR. Vasoconstriction of our systemic arterioles increases peripheral resistance, and it will reduce GFR. But it's reducing GFR so as to return blood pressure back to normal. So the big difference between the previous one, systemic blood pressure and hormonal regulation, and systemic blood pressure and neural regulation, is you'll notice that the neural and the hormonal mechanisms are not worried about returning GFR back to normal. They're worried about returning blood pressure back to normal. So that's what we talked about very early on, where we said the kidney worries about keeping the GFR constant through autoregulation, through myogenic feedback or tubuloglomerular feedback. But when the nerves and the hormones become involved in regulating the GFR, it's not to keep it constant, It's to change it so as to get the blood pressure correct again. Okay? So that's the subtle difference between those types of controlling GFR. One's designed to keep it constant, and the other two, the last two we've looked at, aren't necessarily involved in trying to keep it constant, but change it so as to change blood pressure, because it's the more important thing with these two. Okay? Now, good news for you guys. We've talked for quite a while about one thing, but it makes it a short Camtasia for today. So that's it. Next renal session we'll be talking about that process of tubular reabsorption, that second process that we talked about that happens after we've done all the filtering. Okay, see you in that one. Bye now.